onto our YouTube channel so that those who are, aren't able to wake up to meet this time slot can actually watch it at a later date. So thank you everybody for coming. A warm welcome to everyone across all our different time zones. Thank you so much for taking the time out to be with us today. This is the second of the WSA Seagrass Seminar Series, and this series aims to increase awareness in the lead up to ISBW 14 and WSE 2020 in August next year and held in the United States. Just a few housekeeping rules. Please keep yourselves on mute while the speakers are presenting. We will also be having a Q&A after both presentations. So we'll have Aurora present first, followed by Xiang, and then the Q&A. For the Q&A, if you have any burning questions that you don't want to forget to ask, you may put it in the chat, and I will be looking at the chat and bringing up those questions as and when we have a lull in questions. But if you want to ask your questions live, you're also very welcome to do that. It's nice to interact. And if you are asking the questions, please put on your video, especially if you're doing it live, so that we can see your face and wave hello and say hi and all of the rest of that stuff. I realize we're all starved for interaction because we haven't had an ISBW in 3 years. So without further ado, I shall stop my share here. And we shall start. Okay.
Give me a second to share my screen.
All right, so first up we have Aurora. Dr. Aurora Ricart is currently a postdoc researcher at the Bigelow Laboratory for Ocean Sciences at the Bodega Marine Lab and also at University of California, Davis. It's a bit of a complex situation, but she'll be happy to tell you more about it if you ask her. She is a marine coastal ecologist studying global change in marine systems. Her research addresses questions in 4 main areas, which span seascape ecology, community ecology, ecosystem resilience, and coastal carbon cycling. And today she will be presenting on the topic, can seagrasses turn back the clock on ocean acidification? So without further ado, Aurora, the stage is yours.
Well, hi, everybody. My name is Aurora. I am a postdoc, as Citi said, in 2 different institutions right now, one on the west coast of the United States and one on the east coast. Thank you so much for inviting me today. It's such an honor to be here, and I really hope you enjoy my presentation. So, as you know, one of the main factors contributing to current climate change is the increase in carbon dioxide emissions since the Industrial Revolution. And since then, the carbon dioxide atmospheric levels have increased by 40% due to the burning of fossil fuels and the land use changes, at the point that each year we reach a new record. And the ocean absorbs about 30% of the carbon dioxide that is released into the atmosphere every year. And so as atmospheric levels of carbon dioxide increase, as you can see here in red, the carbon dioxide levels of the surface of the ocean also increase. And this has several consequences for the chemistry of the seawater, resulting in an increased concentration of hydrogen ions, which translate in a decrease in pH. And since the Industrial Revolution, the pH has decreased in average by 0.1 units, with some regions having even higher decreases than that. And so let's briefly review ocean acidification chemistry. Carbon dioxide naturally exchanges between the atmosphere and the ocean and the surface of the ocean. And when it dissolves, part of it reacts with the water to form this carbonic acid, which is a weak acid that at the same time dissociates in hydrogen ions and bicarbonate ion, the same as can be found in baking soda. And as the seawater pH levels, at typical seawater pH levels, some of these hydrogen ions remain as such, but most of them will combine with carbonate ions to form more bicarbonate ions. And so there is a decrease on the pool of carbonate ions, and this has effects over organisms, specifically with calcifying organisms, because the carbonate ion is the essential building block to form calcium carbonates. And so a decrease in this ion difficults the process of calcification. And if waters are undersaturated, then the shells start to dissolve. And here you have one example of many examples that can be found already in the literature, where a theropod has been incubated at carbon dioxide levels expected by the year 2100. And you can see that after 45 days, the dissolution of the shell is very evident. So in summary, chemical changes in seawater resulting from the increased carbon dioxide atmospheric concentrations are multiple and can differentially and interactively affect organisms, marine organisms, with negative consequences for many species. And despite ocean acidification changes the concentration of all dissolved inorganic carbon forms and other parameters, such as saturation state of calcium carbonate, today, for simplicity on this presentation, I will be referring to most of them as pH. So we have a problem, but we might have a potential solution at local scales, which is photosynthesis, that by sequestering carbon dioxide, or in some cases also bicarbonate, can reverse the reactions of ocean acidification. And so photosynthesis will sequester carbon dioxide and will increase oxygen levels and will also increase pH, and respiration will do exactly the opposite. However, primary producers are subjected to cycles of respiration and photosynthesis, which naturally induce the fluctuations in pH through time. And as a consequence of these high frequency fluctuations, which can ameliorate low pH during net photosynthesis, but also exacerbate low pH during net respiration, we really don't know how relevant seagrass ecosystems are for ocean acidification management purposes. And not only that, but in coastal and estuarine areas, there are other factors that can contribute or affect acidity of the water. So in addition to the increase of carbon dioxide atmospheric concentration, we also have riverine inputs, nutrients coming in, eutrophication and algal blooms, and in some regions, upwelling events that bring deep waters enriched in carbon dioxide and also nutrients. And all these processes will increase carbon dioxide production and also can influence alkalinity, which is the ability of the water to resist change for the carbonate system to take up more hydrogen ions, or the ability of the water to buffer changes in acidity. And also, when we move to the seagrass ecosystem, there are other processes that can affect the carbonate system. So here you have a very standard plot that can be found in many carbonate chemistry studies, where alkalinity is on the y-axis and dissolved inorganic carbon form concentrations on the x-axis, and then this color gradient is pH, and you can see different processes here. And for instance, photosynthesis and respiration won't change total alkalinity, but will change dissolved inorganic carbon concentration forms and also pH. But there are other processes, such as redox processes or the dissolution and calcification, that will affect the three of these parameters. So the carbonate system is complex, but luckily we can study it. We can fully constrain all these parameters, and we can do that by measuring at least 2 out of 4 of these parameters that you have here. And we do that either by taking water samples and then analyzing them in the laboratory, but also we can use high frequency autonomous sensors, which will provide nice temporal resolution. And right now we have sensors, autonomous sensors, that can measure either PCO2 and also pH. So looking at what has been done already on this topic, here is an example of one of the first works looking at the seagrass carbonate system, in which they describe large pH dial fluctuations or changes in pH within the same day in summer, reflecting the biological activity of a seagrass bed. And there is also this paper that was published 3 years before, but I am still looking for it. I haven't had the chance to read it. So if any of you happens to have it, please contact me. Decades later, in this other study, they describe a net uptake of carbon dioxide and an increase on the saturation state of calcium carbonate, or omega, in coral reefs that are adjacent to seagrass ecosystems, and especially in periods of high productivity. And this is the first evidence that, the first that I know at least, where they suggest that seagrasses can help calcification in other ecosystems that are close to them. In this other study, they describe increases in pH and increases in omega in the presence of seagrass meadows, but they already start to relate that not only with photosynthesis and respiration, but also with other environmental factors such as water residence time and water depth. In this other study, they relate a habitat trait, or how much biomass of seagrass there is, with the increase of pH fluctuations. And actually what they describe is that submerged aquatic vegetation can alter pH at a whole bay scale. This other study, the first that I know that is using high frequency sensors in seagrass ecosystems, they defined a very strong effect of seagrass leaf area index on the pH and omega patterns. And more recent work, this is a study in which they use also high frequency sensors, but also predictive modeling, in which they describe how seagrasses in current conditions or in high pH will increase pH even more, that will happen here during photosynthesis, but also can exacerbate pH during respiration. But what is interesting from this study is that they predict that decades from now, seagrasses will exacerbate low pH even more badly than now. And lastly, in this other study from last year, what they describe is how acidification in Chesapeake Bay is buffered by a spatially decoupled carbonate mineral cycling. And what they describe is that near-shore submerged aquatic beds, or beds formed by one freshwater angiosperm, Balisner americana, and other and seagrasses, these beds can increase pH in nearshore areas, and that will facilitate calcification of calcium carbonate crystals on the leaves of this species here, which then will be transported to other areas of the bay with higher carbon dioxide levels and more nutrients. And in there, they will dissolve and will buffer this more acidified waters. So this is a very interesting study. And summarizing, there is not too many papers looking at the seagrass carbonate system or looking at how seagrasses can mitigate or influence the seagrass carbonate system, or mitigate acidification. And from the ones we have, we can answer some of these questions, but there is still a lot to be done. So can seagrasses ameliorate low pH? Yes, but also exacerbate it. To which extent amelioration occurs? So there is a lot of variability on how much seagrasses can change pH. What are the mechanisms? It appears that aerobic metabolism or photosynthesis and respiration has a big effect, but also water residence time and water mixing and other redox processes will influence the carbonate system. And so there is still a lot to be done to constrain the magnitudes and timescales of potential buffering effects by seagrass ecosystems. And this is what has been occupying my time in the last years, where I've been trying to contribute to answer these two questions. So can seagrasses ameliorate low pH at relevant ecological and physiological timescales? And does the amelioration vary spatially and temporally? And so in order to answer these questions, we have done different studies. And here This is a study in which we were monitoring seawater carbonate chemistry in different seagrass meadows at a very big scale, temporal and spatial. So we collected pH data inside and outside 7 seagrass meadows of Zostra marina during 6 years in a total of 29 sampling events or deployments. And what we did was to deploy sensors inside seagrass meadows and also outside, 500 meters apart, more or less at the same depth and at the same conditions. And what we observed, looking at all the time series of pH, was that 2 main dominant patterns capture the characteristics of when and how low pH amelioration or exacerbation arise. So we found a positive offset where pH was uniformly higher in seagrass ecosystems, here in green. This is just an schematics of all the time series, and also a similar pH minima in both seagrasses and unvegetated areas with excursions to relatively high pH values during the day in seagrass ecosystems. And as a consequence of these patterns, we observed that overall seagrass meadows exhibited higher average pH in comparison to unvegetated areas where maximal values of pH were higher in seagrasses, but pH minima were similar for both systems. And when we incorporate a time component into this and calculate for each time point of the time series, the difference in pH between seagrasses and non-vegetated areas, or delta pH, what we see is that values above 0, so higher pH within seagrasses and therefore amelioration of low pH, which happens the 65% of the time in our measurements, compared to exacerbation of low pH, which happens less. We also wanted to know the duration and magnitude of events of continuous elevation in pH within seagrass meadows. So what we did was to take that delta pH time series and calculate for particular delta pH thresholds, such as for instance 0.1. What we calculated was how many hours are from here to here and from here to here, and we observed how amelioration events in cold colors in the plot in here are longer and of greater magnitude than opposing cases of reduced pH or exacerbation in this side of the plot. And in fact, in many cases, amelioration events substantially exceeded the duration of a single day-night light cycle, and sustained pH elevations higher than 0.1 units persisted up to 21 days. When looking at the temporal and the spatial variability of pH in seagrasses and unvegetated areas, here you have the pH values, and these are the averages per hour of the day. We observe how amelioration occurs during the day and also during the night. So delta pH is similar in both periods. And also we observed seasonal differences where higher amelioration arise in spring and summer, correspondent with the periods of growth of seagrass meadows in temperate areas. We also observed differences among the sites that we studied, in which north sites actually had better amelioration of low pH compared to southern sites. And we are still looking at what are the mechanisms behind these patterns. But okay, now we have pH patterns, but what happens to organisms? So in order to answer this or contribute in this big topic, what we did was an experiment at the laboratory in which we designed a gradient of seagrass biomass in tanks. We did control the biomass by controlling the soot density, and here you have the tanks outdoors at the Bodega Marine Laboratory, in which we had seagrasses planted in flowering pots. And then we incubated oysters as a model calcifying organisms to know if seagrasses were actually enhancing calcification. And we did that with juvenile oysters that we tracked individually. And we did a second round of the same experiment with adult oysters. So oysters were in these tanks for 5 weeks with constant flowing water, and we measured pH in the tanks and other parameters during the day or at the peak of the sun, and also right before sunrise to characterize respiration or respiration during dark periods. And looking at the pH, what we found is that higher seagrass biomass, you can see here, this is the gradient, induced to higher pH dial fluctuations, which is not surprising. Okay, but when we look at what happens during the day and what happens during the night, what we see that seagrasses increase pH during daytime, that did not decrease pH to the same extent during nighttime, as you can see here. We were actually expecting to see more exacerbation or more decrease in pH with higher seagrass biomass, and this didn't happen. So when looking at the oysters, this is relative shell growth of the oysters. Here is the same seagrass gradient, and what we see is that shell growth was increased with seagrass biomass in both juveniles and adults. But that experiment was done in ambient conditions of seawater. And we also wanted to know what could happen if we elevated carbon dioxide levels in seawater. What would happen with this interaction oyster-seagrass? So we did another experiment in which we incubated oysters and seagrasses, but in this case in chambers, enclosed chambers. These incubations were only done for 3 hours, in which we measured calcification in a different way, using a different technique. So we have oysters alone and oysters and seagrasses in the chambers, and we did incubations during light exposure and also dark periods. And this work was led by Sarah Merola at the Bodega Marine Laboratory. And what we found, you have calcification rates here on the y-axis. These are the oysters alone. These two are the oysters and seagrasses. Light periods on the left, dark periods on the right, and then clear blue is ambient CO2 conditions, dark blue elevated CO2. What we found is that seagrasses enhanced oyster calcification in both ambient and elevated CO2 conditions during exposure to light. But more interestingly, what we saw is that there were no differences during the dark periods. So there wasn't exacerbation of low pH because of respiration of the seagrass when oysters were incubated in the seagrasses. We didn't observe higher dissolution of the shells. So the dissolution was actually the same for all treatments. So take-home messages from all what I said. For you, for the future, seagrasses elevate pH compared to non-vegetated areas for extended durations of time. We observed important seasonal and geographical patterns modulating this trend, and there are implications for organisms. Some management outcomes. Seagrasses can modulate the carbonate system, but the scale of that effect is local, it's very small, and the temporal scale will go from hours to weeks, depending on many different factors. So seagrasses can contribute to increase adaptive capacity in coastal areas, but there, of course, there is still a lot to be done in order to use them for management purposes regarding ocean acidification. So current gaps in knowledge and directions of potential future work on this topic. There is a need to understand the relative importance of all the mechanisms influencing the seagrass carbonate system. And there is also a big gap on studies looking at alkalinity fluxes in seagrass meadows. Also, we need to take into account that the carbonate system can be decoupled, and so that adds a little bit more complication into these studies to characterize what is really going on. And there is this work that is right now in review, but the preprint is available, and I think they do a very nice description, or this is a very good example of how the carbonate system can be decoupled in some cases. And of course, if we want to generalize these results or other results, we need more studies in other seagrass species in other regions. And one important thing to consider is that we need more field studies considering other community interactions, such as, for instance, predation, to know what are the net outcomes of all this. Because, okay, seagrasses can enhance calcification, but if predators are eating those oysters or those bivalves, that the net outcome is going to be It's going to be 0. So there are many studies done in the lab, but there is still a lot to do in the field in order to predict what will happen. And related with that, there is a lot of ocean acidification research looking at the impacts of ocean acidification, but there is not too much done looking at potential solutions. So a lot of work to do. And I like to end with this question, in which I ask if the amelioration that we observed currently in the field and also in manipulative experiments in the lab, if that will hold in a future world with high CO2 and high temperatures. So climate change mitigation strategies are mostly based in these two different aspects: reduction of greenhouse gases emissions and increase of greenhouse gases sequestration, and seagrasses can help on these ends. But the only known realistic option, the only known realistic mitigation option at the global scale is to reduce what we are emitting. And so with that, I would like to acknowledge or to thank all my collaborators and funding sources. I also want to acknowledge that this research was developed at the homelands of numerous people whose cultures and relation with the ocean was and still is disrupted. And thank you so much for listening. I would love to take any questions.
Thank you, Aurora. That was great. Very informative, looking at it at the macro scale, which is great. We will hold the questions till after Xiang's talk, if that's all right. If you do have any questions in the meantime, please feel free to put it down in the chat and I'll take note of it and we will get back to that during the Q&A session. Thank you, Aurora. Okay, so now we've looked at it at a macro scale. I think we want to move over to Xiang. So Dr. Xiang, our next speaker, is a research fellow at the St. John's Island National Marine Laboratory. What a mouthful. And the Tropical Institute of Marine Science at National University of Singapore. So her research interests lie in examining how marine phototrophs, specifically seagrasses and corals, respond and adapt to changes in their environment due to climate change and water quality. And today, Xiang will be looking at the micro or plant scale. And she's asking the all-important question, especially for us seagrass people, whether seagrasses can be the winner in the scenario of ocean acidification. Xiang, over to you.
Hey, thank you, Siti, for the introduction. Let me share my screen. Okay, awesome. Okay, good morning and good evening to everyone who turned out for the talk. It was awesome talking to seagrass people again amidst this weird situation we are all in now. Okay, so previously Aurora was asking the very important questions like whether seagrasses, whether the pH buffering potential of seagrasses, will it still be around in a future world where there's high pH, low pH and high temperature. So what I'll do is to kind of share on my research of what I've done and try to answer whether seagrasses can actually be a winner under ocean acidification. Okay, so this theory is kind of suggested by the fact that seagrasses are thought to be carbon limited and they have a higher affinity, higher preference for using carbon dioxide, dissolved carbon dioxide in seawater compared to bicarbonate that's present in a greater quantity in seawater. So under ocean acidification, there's a predicted increase in dissolved inorganic carbon, with especially a high, more than 250% increase in carbon dioxide, dissolved carbon dioxide that's available for seagrasses. So in short, basically people think that it will be a buffet for seagrasses when pH drops and seagrasses will thrive. And there have been very, very visual evidences of seagrasses thriving in a future world of ocean acidification. So this picture is taken at near CO2 vents in Papua New Guinea. And what you see here are bubbles of carbon dioxide streaming from below ground. And you see a large seagrass meadow thriving all over. And this is a very beautiful sight. And comparing seagrasses and other biota that's living around these CO2 vents, seagrasses have been indicated to be a winner under ocean acidification. So there have been a number of research that has shown that seagrasses do respond positively to carbon dioxide enrichment. My research included in that primary production and growth rates of various tropical seagrass species responded positively. They grew more, they photosynthesized more with more CO2 given to them. And this is very good news because we all know that primary productivity is a cornerstone of all the important ecosystem services that seagrass provide to the world, especially in times when people are becoming more and more aware of the effects of climate change. The ecosystem services of carbon sequestration and storage, shoreline stability, all these services that has always been kind of outside of mind for people suddenly became a buzzword among a lot of funders, a lot of lay public too. However, as practitioners, as scientists who work on seagrass beds, all of us know that there are also many factors that can affect seagrasses that can stop them from being that productive, such as eutrophication, reduction in light availability due to sedimentation, or even when a marine heat wave hits, as we have seen repeatedly over the past decade even, when a marine heat wave hits, basically an entire bed of seagrass meadow just gets wiped out. And all this would essentially put a stop to how much good seagrasses can do for the world. So my research just wants to ask, like, okay, so in a world where there is more CO2, yes, we know seagrasses probably do better. They're able to utilize that extra carbon dioxide for photosynthesis. How does that get supported or not supported by other co-occurring environmental stresses, like reduction or increase in nutrients in seawater or even temperature rise? So this is my little nice, very cartoony understanding overview of the internal workings of seagrass. And I'll go step by step to show what I've done. So let's look at nitrogen availability. So nitrogen is important for carbon assimilation into many organic nitrogen-containing compounds. And nitrate and nitrogen metabolites have been indicated for their regulatory and signaling roles in carbon metabolism, such as photosynthesis, starch accumulation, and even organic acid synthesis. So essentially, nitrogen is very important to sequester carbon away from carbon cycle so that the accumulation of sucrose does not inhibit that carbon cycle in the seagrass. So there are studies done on terrestrial plants which show that the initial increase in photosynthesis due to higher CO2 levels, that increase eventually plateaus off and declines as when the plant becomes nitrogen limited. So then I kind of asked, like, so will giving extra nitrogen in the form of nitrates be beneficial for seagrasses? Maybe it will help them to take up more carbon dioxide and boost productivity. So in this study, actually most of my studies presented here are done in Australia, Great Barrier Reef, where I did my PhD. So in this study, where I worked at a mid-shell reef, where nitrogen levels in seawater are not that high. Nitrate levels in seawater is not that high. What I found was quite surprising, because it seems like even though the seagrasses do respond to CO2 enrichment, they do not necessarily require that extra nitrogen boost to be able to utilize that the extra carbon that's provided. Okay, so then next I'm moving on to light. So upstream of carbon cycle, the carbon cycle, the availability of light would affect how much carbon gets sequestered, because light provides energy for fixation, and it also provides energy for the uptake of bicarbonate. So there have been studies also to show that seagrasses, they can only respond to carbon dioxide enrichment when there is adequate light provided to them. So this has been done for temperate species, mostly temperate species. And there's one done in East Africa, tropical species, ovalis. What this study showed was that for seagrasses that's kind of adapted to lower light levels, that increase, that boost in carbon dioxide, that boost in dissolved inorganic carbon, actually benefited plants that have been not the low light. That's a word I triple. Okay, so that extra boost in dissolved inorganic carbon is more beneficial for seagrasses that are low light adapted. Okay, so what I found was that for Simoldosia cerulata and Halodiri uninervis, light seems to be the very predominant driver of productivity in both species, such that basically the effect of carbon dioxide on productivity is not very, very obvious. The only really interesting thing that I found was that for Simoldosia that is living in low light conditions, basically under subsaturating irradiances, growth rates were actually boosted by an increase in carbon dioxide provided. But however, for other effects, we don't really see, we see more of a light effect than carbon dioxide effect. Okay, then moving on. So with ocean acidification, there comes ocean warming. They come hand in hand together because, well, the root cause is the same. So temperature is a very big regulatory of carbon metabolism in influencing processes such as translocation, respiration, fixation, and storage. And eventually all this affects the growth of the seagrass plant. So true to what I expected, temperature plays a very big role in regulating primary production and growth rates in both Simoldosia cerulata and Halodiri uninervis. So much so that it seems like carbon dioxide availability is not really affecting, so much so that it plays a much bigger role than carbon dioxide availability in terms of regulating carbon metabolism. And yeah, so the temperatures that I've tested in this study falls within the thermal ranges of the seagrasses here. So within a seasonal variation, when it comes to summertime, where temperatures are higher, seagrasses are better able to utilize the extra carbon dioxide available for them to increase their growth rates and productivity. But however, we also know that there are sometimes, more frequently now, where temperatures tend to tip over that sweet spot, that thermal limit. So when that happens, catastrophe strikes, I would say. So when temperatures are warm and just nice, like at the thermal limit and with extra carbon, seagrasses tested are able to increase the productivity slightly, positively. However, when they're under a very stressful condition, so conditions that are akin to somewhat of a heat wave, heat stress, productivity drops drastically with like 4-4, 24, that kind of range. So yeah, so that is kind of bad news for us, because in a world where there's high CO2 and high temperatures, seagrasses don't seem to do so well. So, okay, so just to kind of summarize the results so far, because I think I've shared quite a bit. Tropical seagrasses can and potentially can be winners under ocean acidification only if these conditions are met. So what I found is that light availability and temperatures are very strong drivers of carbon metabolism, much more so than the availability of carbon dioxide or even dissolved inorganic carbon. So for seagrasses to be able to utilize that carbon under ocean acidification, they need adequate light that is above their saturating irradiance requirement. They also need temperatures that are within their thermal tolerance limits. And also, will also require seawater nutrient levels to be low, because even though we know that seagrasses don't really, might not really require all the extra nitrogen for them to boost their productivity under ocean acidification, we do know there's algae, there's also other macrophytes out there that would really take advantage of increased carbon and nitrogen availability in seawater. And that might lead to competing microalgae to grow in lieu of seagrass beds. And what my work has shown consistently is that there are species-specific responses throughout. So even for the same species that I've collected from different sites within Australia itself, they do show different degrees and different dichotomy in whether they respond to carbon dioxide enrichment or not. So that puzzled me a lot, and that led me asking questions such as, does the geochemical properties of the site affect, especially geochemical properties of the sediments, affects the ability of seagrasses to respond to ocean acidification? Is there a population differences? I mean, I would expect a population differences that's going to affect whether seagrasses can respond to carbon dioxide enrichment. And then, as what Aurora say, there's very little known about the community interactions within seagrass beds itself. How does that interaction sustain or be altered under ocean acidification, and how does that affect seagrasses in terms of the ecosystem services they provide? We do not know that yet. So just a final wrap up. So I get asked these questions a lot, like, so what if seagrasses are winners in ocean acidification? What does that mean? So a lot of potential funders, a lot of colleagues ask me, why are seagrasses important essentially? So usually what I tell them is to save the corals. So, because, well, they're kind of more important, but no, that's not true. So if seagrasses are winners in ocean acidification, we will know that they are able to provide a lot more of the ecosystem services, such as carbon sequestration, storage, and providing shoreline stability. And there's also one that Aurora described just now, which is the potential to buffer pH changes in seawater, and in turn that might benefit downstream calcifying organisms, such as coral reefs that everyone cares about. And this can potentially form a feedback system where the reef increase their accretion and provide more sheltering benefits for seagrasses that's at the back end, depending on their orientation. So I thought this would be a very interesting prospect to explore, especially in Singapore, where I'm based now. So firstly, Singapore is quite awesome because despite our tiny size, we still have seagrass beds. And most of them, even though they are tiny pine size, they are all in vicinity of habitats that have calcifying organisms. And secondly, Singapore is also very interesting because if you see, oh yes, we are here. We are very tiny in the sense of a world map. So unlike many other countries like the US or Australia, where your land is large enough for water quality management to actually have an effect, to have potential effect on bettering your seagrass beds. We are kind of surrounded by huge land masses and huge water bodies. So there's the influence of both oceans, in the Indian Ocean and Pacific Ocean, on our seawater. And with that, they bring along whatever comes off from the land, from whatever land use there is from our neighbors. So for example, during the southwest monsoon, which is I think now-ish to September, okay, depends. But basically during the southwest monsoon in the middle of the year, we have waters bringing in humic acid rich waters coming from the peninsula of Malaysia and also Sumatra. And it brings a kind of pH low water to the seagrass beds in Singapore. And when the season reverses, during the northeast on monsoon, we have influence from the big sea, the big pond called Pacific Ocean, that's bringing a lot of seawater to seagrasses in Singapore. And so basically, essentially, we are kind of a sitting duck, I don't know, to the different influences that's coming beyond our control, I would say. Okay. And lastly, Singapore is interesting because if you look at the map of Singapore itself, the coastlines are very, very straight, which is not what nature would have shaped things out to be. So a lot of our shorelines are man-made or have been developed. So have been developed and artificially armored. So the seagrass beds essentially they are kind of blocked off by seawalls. They cannot really migrate. There's nowhere for them to go inland, and further out there's a lot of reclamation work, dredging, shipping traffic, and they can't really move out either. So they're kind of trapped. So these are all the different influences we have in Singapore that makes the place so unique. Okay, so saying all that, we have we have a project currently that's still ongoing that's looking at the potential of seagrasses to mitigate ocean acidification effects for downstream coral reefs at such a small scale that we have. So one of the premises that we have to address is whether seagrasses in Singapore, after being bathed in low pH waters and high pH waters periodically, how would they respond to having more, having higher temperatures and having more carbon dioxide? So thankfully, I mean, we managed to get this premise addressed in that seagrasses do better under under climate change conditions in Singapore for this species, Pseudomonas cerulata. So yes, seagrasses can be potential winners in Singapore waters. And we also measured pH changes in tanks itself to show that the pH changes in closed chambers, they do track the variation in dissolved oxygen quite well. But this is only under very low flow rates. So what could be done further was to do more field surveys to measure the various seawater chemistry parameters and do a hydrodynamic modeling of such a small scale to see whether the seawater does that altered seawater with that water with altered chemistry, does it get transported out of the seagrass bed to downstream coral reefs? And by the time it reaches coral reefs, how much of that benefit still remains for the corals to calcify? So yes, so with that, because it's still work in progress, I would love to share with you more once we have more results. But now I'd just like to thank my sponsors for making sure I still have a job to do all this. Hopefully they will continue to fund me for the next few years. Okay, thank you.
Thanks, Xiang. Thank you, Xiang. All right, so we have gone through both presentations, so now is the time for you guys out there to participate. So if you have any questions, please feel free to raise your hand or type it in the chat, and I will call on you. So does anybody have a question that they would like to ask live now? Don't all rush at once, guys. Like, okay, no? Anyone? Oh, okay. 2 participants have raised their hands. Now, let me see. How do I? I cannot see who has raised their hands and who hasn't. Why is that? Okay, we have a question from Caitlin and from Denise. So let's go with Caitlin first.
Thank you so much. Those were excellent talks. I really enjoyed both of them. My question is for Aurora. Given in your paper, I think you found one of the patterns is that you have a higher fluctuation. So there might be that more dynamic change in pH throughout the day.
Oh,
Oops, Caitlin's having. Caitlin,
yeah.
you've kind of
You
frozen.
froze.
Okay, Caitlin, when you come back online, maybe type your question in the chat. We can go to Denise first if.
Sure. Hi, you can see me. Hello. Thank you so much to both speakers for both the presentations. It was so engaging. My question is for Aurora. Aurora, I think in your last slide, you showed that there was no exacerbation. Is it of pH amelioration under enhanced CO2 concentrations? I was just wondering, what are your hypotheses or guesses as to why there was no exacerbation?
That's the 1 million question, actually. I mean, I have guesses, I can speculate, but I don't really have an answer or evidence to say this is what's going on. My guess is that respiration rates are so low to make an effect on the system, on the carbonate system, which is what we are looking at. We didn't measure respiration rates or photosynthetic rates on the seagrasses. We just looked at the water chemistry. And my guess is that respiration rates in those waters where we work is so cold that are low. And that doesn't make a big change on the chemistry. Of course, there's change. but the photosynthetic change is bigger. Anyway, this is all speculation. There also might be, well, those chambers were enclosed, so there's no escape. But for the other experiment that I presented, where we saw smaller decreases in pH during the night, we were using big tanks with open tops, and there can be an effect of outgassing, and that can explain some of the low effects that we see during dark periods or at night. But yeah, I mean, I am very intrigued by those results. So if any of you have ideas, discussion, I welcome you to contact me. I mean, we can discuss now or contact me later, because I will love to discuss all this.
So, thank you.
Thanks, Denise. So Caitlin has indicated that her question is quite similar, so we'll move on to Heidi.
Cool, thanks. Great talks, you guys. I really enjoyed both of those, and great questions so far too. That was actually one of the questions I have when reading that paper, and I'm curious what everyone comes up with. But my question was kind of related to what Yen said at the end of her talk about the hydrodynamic modeling and the importance of flow, which I'm actually then going to relate it back to Aurora's data, because I know that you talked a little bit about water height, I think, in your paper, but did you, and today you showed some of the seasonal data, but did you see any relationship between the pH changes and like depth or tidal phase, and the like degree of that magnitude and change? I know that was something that came up in the like Cowick et al. Modeling paper for similar regions.
I don't know if I mean, we can both actually answer that question. I don't know if you were asking Jan or me. We did measure some, not some, we did measure currents or flow rates. And also we were using ADVs, sensors that can provide flow rates, but also some turbulence data or hydrodynamic parameters of what's going on. And what we observed was that definitely more turbulence was decreasing the effect of the seagrass, or was decreasing amelioration. So pH was not changing that much when the turbulence was increasing. So that was very, very clear. And with the tides, I have to go back and look because I really can't tell right now, but with the depth, there was a very clear effect that water depth is decreasing the potential of amelioration, which can be because of the tides flowing, or can be because of the light. I mean, water depth will limit light arriving. So that's both effects there that I can't discern right now, but wait a little bit more. There will be results on that end.
Xiang, do you want to pipe in here before we go to Paul's question?
Okay, so for us, right now we are using data for water height, for water flow from calibrated models, because we were going to put out sensors, and then there's COVID, so we took back our sensors. So yeah, it was a very eventful project anyway. So yes, I do agree that water height, looking at the flow, looking at turbulence is very important in this hydrodynamic modeling, especially for Singapore itself, because the scales are so tiny. The boundaries are, setting the boundary conditions are really hard itself. And on top of that, we have ship wakes. We have crazy ship traffic in our shipping lanes, and that creates a lot more mixing and turbulence. So that's why I thought it'd be very interesting to see how all these small scale coastal water motion will actually affect the transport of seagrass water to coral reefs, downstream coral reefs. yeah, so work in progress too.
Thanks, Xiang.
Sorry.
Was that the end of your question, Heidi? Can we move on to Paul? Okay, cool. Thank you. Hi, Paul. How are you?
Yes,
I have a
hello.
question.
Thank you for the two awesome presentations. It was very interesting, both Jan's and Aurora's. As typical of research and science in general is that you end up with more questions at the end than at the beginning. I had hoped that we would have the perfect answer. Yes, it's all going to be good. More seagrass will help to ameliorate acidification, but I don't think we have a simple answer here. I have a question for Aurora. I really liked your experiment with the oyster juveniles and seagrass together in cylinders and looking at the net effect. I couldn't help but wonder, there's one very significant component in that experiment that is missing, and that is what's happening in the sediment, because you have the individual seagrass plants and the oyster juveniles, and you see the interaction, but that shows, of course, what the plant in itself is doing to the whole chemistry and balance of it all. But it doesn't really include what's happening in the sediment. And as we know, seagrass meadows at the ecosystem scale, they trap a lot of organic material and they bury it. And so they tend to be more rich, the sediments under seagrass, than the unvegetated sediments. And therefore, obviously, their respiration rates is much higher as well at night. So what happens at night has a lot to do also with the net balance between the respiration and photosynthesis. Yeah, I just wonder if you have thought about doing the same type of experiment with the juvenile oysters in the field, in the real seagrass ecosystem, where you have that sediment component included, and then maybe you can compare the two results and say a little bit more about what's happening in the sediment, how that contributes to it all. Thanks.
Thank you for that comment. I mean, you are totally right. This is a laboratory experiment, and things in nature are more and more complex than that, definitely. One thing that I'm going to say on my favor is that we used sediments on the tanks, but of course that's not mimicking the natural system, but the seagrasses were planted in pots with natural sediments in all the tanks, even if we didn't had seagrasses on the tanks, like the control tanks that were only water, there were sediments. So trying to put the same amount of sediments in all the tanks, trying to mimic as much as possible natural conditions, which is of course not the same. And there's that question, like what is going to happen in the field in a seagrass gradient or seagrass biomass gradient in the field. And I am not doing that work, but I have colleagues that are working on that. They actually have outplanted oysters, juvenile oysters. I didn't mention those were 2 to 3 months old oysters. So they have been outplanting them inside seagrass meadows and in gradient from the inside of the meadow to the outside, and also in non-vegetated areas to look at this. So hopefully these results will come out soon, and that answer will be, that question will be answered.
Awesome.
But it's a great comment. I mean, definitely lab experiments are lab experiments. We cannot generalize from only that.
Thank you.
Thanks, Paul, for your question. I think Nok has a question next. Nok?
Hi, both of you. I actually have a question for both. I've been really fascinated by intraspecific variations lately. And since you both working in physiology and also Xiang working in Australia and Singapore, so I was wondering if you have noticed much of that, because, well, this is important for future prediction, and whether certain population is going to be have a bit more of adaptive capacity, or the others will be more sensitive. Any comments on that? Thank you.
Xiang, maybe you want to go first and then Aurora.
Okay. Hello, Nook. Good morning. Okay, so there definitely has been a lot of interest-specific variation, so much so that I started to doubt whether I'm a good scientist because I keep getting different results on the same species. But then I realized eventually that, you know, different populations, well, what do you expect? They would respond differently because they have been bathed in a different environment their whole lives. I mean, seagrasses don't have passport to cross borders anyway. So yes, so I'm very interested to maybe look at reciprocal studies that, yeah, I think we have discussed before, Siti. Looking at how different populations respond to similar stress or similar boosters of productivity. And yeah, and because populations can differ so much in their responses, not just to carbon dioxide, but also to temperature and to light. I mean, we have lots of this evidence in literature. As to what you say for predictive modeling, it'd be important to elucidate like how different populations respond, because if we need to do monitoring, if we need to do preemptive environmental interventions, we need really accurate data that is specific to the site itself. So yes, I think even though we work on the same species, there's very good motivation to just compare results of similar experiments, just to look at what's causing that difference. Yeah. Okay.
Can I comment? Yeah, I think, I mean, there is a lot of evidence to, a lot of evidence saying that there is intra-specific variation in organisms, in species, sorry, and I think that will also be for seagrasses, and that either can be because of environmental factors on the particular site or the site context, but also genetics. What are the genetics? What is the genetic information of that particular individual? And where you were suggesting that it's super important in order to do predictive modeling or to know what will happen in the future. So I just wanted to comment that we, in one of the studies that I presented, the sites, the one that was looking at different sites, different seagrass meadows, is the same species, but the behavior of the meadow is so different from one site to the other. And I mean, we haven't looked at what exactly is explaining that, but it might be environmental factors that are different or site context things, but also differences on the genetics and the adaptation of those individuals to the site. So that is a thing to look at in the future, for sure.
Thank you, Aurora. Okay, so I think we have to wrap it up because we are 12 minutes over, but clearly this discussion is a good one, and you're free to, contact the speakers or each other to further discussion. In fact, Xiang, I think we need to talk after this. But anyway, so thank you all for coming. As I said, this will be recorded, so if you want to view it again in great detail, and relive this experience, you're free to do that once we put it on our YouTube channel. So I'd like to wrap it up by thanking our two speakers, Aurora and Xiang. Thank you so much for sharing, your studies and your thoughts with us today, and also for the audience for the great questions. And I hope that we can keep this discussion going, for when we are able to see each other again at ISBW. Just a couple of quick other announcements. There will be a student event that we are planning to host. More details on that coming up later. And if you do have any questions, after this session for either Xiang or Aurora, please feel free to email us, and then we will be able to forward it to them. All right, thank you very much, everybody. So let's conclude that. and have a great day ahead or evening ahead. Thank you. Bye.
Thank you, Siti. Thank you,
Thanks,
Aurora.
Yanxiang. Thanks, Aurora.
Right.
Thank you,
Bye,
everyone.
Bye, everybody. It's nice
Bye.
to see you.
Yes, it was really nice to see you guys
Yeah.
again.
Yeah.
All right, bye.